Hello everyone, welcome to Infinity IAS. I am Dr. Naresh and today we will be looking into an important topic for science and technology, that is CRISPR CAS system. This topic is not only important for prelims as well as for mains. This year in mains we have seen that there was a question asked on blue LEDs. It was awarded Nobel Prize in the 1990s. So here we see a pattern that any invention or discovery that has been awarded Nobel Prize has become important, particularly in GS3 paper of the mains examination. So if we see, CRISPR CAS system was awarded Nobel Prize in chemistry, that too for 2 women scientists, which makes it even more important. So what is CRISPR CAS system? CRISPR CAS system is a gene editing tool. CRISPR CAS system is used to edit genetic material in any animal or human cell. So, what is CRISPR CAS? CRISPR stands for clustered, regularly interspaced palindromic repeats. That means it is a genetic material. It can be RNA that is used to target certain genetic material in the animal cell. CAS means CRISPR associated protein. So this is a protein, this is RNA. So if we look into the mechanism of action as to how CRISPR CAS helps in gene editing, we can look at it as CRISPR is used to identify the target DNA sequence that we want to cut in the animal cell, and CAS protein acts as the molecular scissors. That means CAS protein actually cuts the DNA that has been identified by the CRISPR sequence. So if we see in the DNA, we have double-stranded sequence of bases. So CRISPR will have a certain gene that will help in identifying the target. So for example, if we want to cut the DNA here, the CRISPR will have this sequence. So when the CRISPR CAS system goes into the animal cell, it identifies this and gets attached here. Now in the CRISPR CAS system, the CAS protein will cut the DNA here. So CRISPR helps in identifying and CAS helps in cutting the DNA at that specific point. Now there are many types of CAS proteins, almost 13 types and many subtypes among that. But the most commonly used one is CRISPR Cas9 protein. Cas9 protein is the most commonly used in laboratories. Now, what is the origin of CRISPR Cas system? CRISPR Cas system was originally the immune system of bacteria and other old organisms, single-celled organisms, such that when a bacteria gets infected by a virus, bacteria uses CRISPR Cas system inside the bacterial cell to damage this viral organism that is entering the bacteria. So this is originally, CRISPR Cas is originally the immune system of bacteria and other prokaryotic cells. Bacteria and other prokaryotes. So it is the immune system of bacteria and prokaryotes. We have taken this out of these cells and we are using it in laboratories to artificially edit the genes in the animal cells or human cells. So what can CRISPR be used in our laboratories or for human advantage? So CRISPR system can be used to add remove or even alter specific DNA. So, for example, we have a DNA in a human cell or an animal cell. So, if we want to add a specific gene into this genome, so we can use CRISPR to cut the gene here using the target and the Cas protein. Then we can add whatever gene we want to this specific genome. The same way to remove, if we want to remove this specific gene in the whole genome, we can use CRISPR to cut here as well as here and then attach these two ends so that this gene is removed from the genome. The same way we can alter specific DNA as well using CRISPR. CRISPR can be used to cut the genome and then specific sequences whatever we want we can add or change the specific bases so that the gene can be expressed in the way that we want. So it can be used to add, remove or even alter DNA. So this helps CRISPR Cas system to be used in biology, particularly molecular biology and genetics, in medicine and in agriculture. So if we look into applications of CRISPR Cas system in agriculture, it can be used to increase plant yield, quality, disease resistance, herbicide resistance, etc. How can this be done in plants? For example, a plant genome can be cut at specific sites using CRISPR and specific genes can be added such as a disease resistant gene or a herbicide resistant gene, or we can even increase the yield by inserting whatever the genes that we want into this plant genome. For example, herbicide resistance. What is herbicide resistance? All the plants that are grown in farms will have weeds that will be grown in between them. These weeds will be destroyed by weedicides and herbicides. But if this weedicide or herbicide also kills our plant, then we will lose the yield. So if we insert a specific herbicide resistance gene into this plant, we can use this chemical herbicide or a weedicide to kill only these weeds and not our plant. In this way, our farm yield will increase. At the same time, the problem of weeds will decrease. So such things can be done using CRISPR in agriculture. When it comes to medical aspect, CRISPR has been used to diagnose, treat, as well as monitor the treatment in case of various diseases. For example, Zika virus. CRISPR is used to diagnose the different strains of Zika virus. For example, the African strain as well as the American strain. CRISPR is being used to diagnose and differentiate between African and American strains of Zika virus. The same way, it can be used to treat various diseases. For example, there are genetic diseases such as DMD that are caused due to the mutations in certain genes in the human genome. We can use CRISPR to alter those specific genes to treat those diseases. And then when it comes to research, it is used in the field of genetics and drug discovery. This is still progressing and there are many more avenues where CRISPR can be used, particularly in cases of drug discovery. So when it comes to prelims, we have to know what is the mechanism of action, what are the uses and applications of CRISPR, and also what is the regulatory mechanism in India when it comes to gene editing. In India, gene editing comes under rules for the manufacture, use, import, export and storage of hazardous microorganisms or genetically engineered organisms or cells, 1989. These rules have the regulations that administer the genetically engineered organisms. This is notified under Environment Protection Act 2018. So for prelims, this is important. Environment Protection Act 2018 has the rules regarding, and then there is also National Ethical Guidelines for Biomedical and Health Research involving human participants by ICMR, Indian Council of Medical Research. For prelims, this is important. It has the National Ethical Guidelines for Biomedical and Health Research under ICMR. So, what is actually mentioned in these guidelines? It is mentioned about modification, deletion or removal of parts of heritable material. This is what is exactly mentioned in these guidelines. There is no mention of genomic editing. There is no mention of genomic editing in these guidelines, but it is mentioned that modification, deletion or removal of parts of heritable material. So these are the points that are important for prelims. In the next part, we will look into what are the important points when it comes to CRISPR CAS, when it comes to the mains examination. Thank you.